Ekaterina. Now is the turn of Ekaterina Bayurova. She is from the Chumakov Federal Scientific Center for Research and Development on Immune and Biological Products. He's from Moscow, Russia. And the title of her talk is HIVE-1 reverse transcriptase expressed in epithelial tumor cells enhance their tumorigenic and metastatic potential through promotion of EMT and stimulation of mitochondrial respiration. Please go ahead.
Do you hear me now?
Yes, we can hear you.
And can you see my slides?
Yes, now, yes.
Yes, it's good. Hello, everyone. My name is Ekaterina Bayurova, and I want to present my work on the topic of expression of HIV reverse transcriptase and its involvement in mitochondrial respiration and motility of epithelial cells. As many of you may know that people living with HIV are at increased risk of developing cancers, and previous years it was linked to the depletion of CD40 cell count, but nowadays in the era of antiretroviral therapy, the level of CD40 cells in HIV positive people is the same as in healthy population, so it's not be the case. However, the level of AIDS-defining cancers is total fourfold elevated compared to a healthy population, and for example, for Kaposi sarcoma, incidence level is 800 times elevated compared to healthy population. And not only AIDS-defining cancer, but also cancers that were not earlier associated with AIDS also have an increased incidence rate. For example, cancers associated with viral infections such as hepatitis B and hepatitis C viruses, or human papillomaviral infection, and brain cancer in the list of other cancers that is not previously been associated with HIV is also elevated in people living with HIV. So what was the mechanism, what was the point? And for some of HIV proteins, it was shown the direct oncogenic properties. And you can see the list. For example, for NOF, it was described that it promotes tumor growth, that is associated with epithelial to mesenchymal transition and tumor proliferation. GP120 stimulates glycolysis and epithelial to mesenchymal transition. All these proteins induce ROS, and we in our team shows that reverse transcriptase also promotes tumor growth, and it also induces ROS, reactive oxygen species. So we would like to go deeper and evaluate the effect of HIV reverse transcriptase on the metabolic activity and motility of tumor cells. For that purpose, we developed a surrogate model. We used 41-Luc2 murine adenocarcinoma cell lines, which express luciferase and synthetic gene encoding for consensus sequences of reverse transcriptase clade A. It was made from the sequences of patients, treatment-naive patients from former Soviet Union countries. And we introduced with the 41-Luc2 cells with lentiviral coding for this RTA consensus with different multiplicities of infections, and for the cells were single cell clones, and we generated 3 different clones named RT1.3, 5.3, and 20.1. We evaluate gene copy number in these cell lines, and we used the digital droplet PCR for that purpose, and for 1.3, the copy number is 0.3, and for 5.3, 2 times higher, and for subclone 20.1, unfortunately, we have no data. That's why we decided to focus on protein expression, and we evaluate the protein expression level by Western blot using our antibodies that we previously developed against reverse transcriptase of clade A HIV. And as you can see here, all the subclones express reverse transcriptase at different level, as the level of expression correlates with the number of insertions and the number of the multiplicity of infection number used for antiviral transduction. We evaluated the level of reactive oxygen species in all generated subclones, and we also decided to compare our parental 41-Luc2 cells to non-transformed murine NH3T3 cells, and we find out that 41-Luc2 cells as the tumor cells have increased basal level of reactive oxygen species compared to non-transformed cells, and expression of reverse transcriptase induced reactive oxygen species hyperproduction in all the subclones, and this hyperproduction was also correlated to the level of level of protein reverse transcriptase per cell. And we used several antioxidants to quench this reactive oxygen species, and we used tocopherol and N-acetylcysteine, and both lead to decreased level of reactive oxygen species. However, the level was still higher than in non-transformed murine cells, NH3T3. We analyzed migration capacity of all generated subclones. We used classic wound healing assays for the purposes, and the cells were analyzed the time period from 1 to 14 hours and from 14 to 18 hours, because the doubling time for this subclones is about 15 hours. So we assume that during the first 14 hours, there was no doubling and only migration. As you can see here, that reverse transcriptase expression lead to increase in migration rate in both in all subclones, and this increase was correlated to the level of reverse transcriptase expression. And what's more interesting, that at the late stages, when wound was almost closed, parental 41-Luc2 cells almost stop the migration, and all the RT expressing subclones remain the same level of migration rate. So they all lost the contact inhibition and all the stop factors. And we also noticed that for some cells, they changed migration phenotype from epithelial migration when they migrate as a sheet to mesenchymal type of migration when they migrate as a single cells. And there is an example of the wound with a single cell in the middle of the gap that are marked in with black arrows on slide. So we decided to analyze epithelial markers of epithelial to mesenchymal transitions. We assessed by PCR the level of Ekaterina, Ekaterina, vimentin, twist, and snail. And as you can see, the level of all the mesenchymal markers increased with a increasing level of protein of reverse transcriptase expression per cell, and relative level of twist expression highly correlated with the level of reverse transcriptase protein per cell. And for Ekaterina and vimentin, their level correlated with the level of reactive oxygen species measured in this cell. So in vitro is in vitro and in vivo is in vivo. So we analyzed the tumorigenic and metastatic progresses of the generated subclones. For that purpose, we inject them into the base of the tail vein, into the back of the tail in urine in BALP-C mice, which are syngenic mice, and we analyzed tumor formation by monitoring of bioluminescence signal from the site of injection, as 41-Luc2 cells express luciferase as a reported gene, and also by physical assessment of tumor volume at the end of the experiment. As you can see here is the tumor growth rate represented by the bioluminescence signal from all reverse transcriptase expressing SUB clones was higher than for parental cell line starting from day 5 until the end of experiment, and tumor volume was correlated to the level of twist expression in the cell lines, and total flux bioluminescence signal from the site of injection correlated with level of reverse transcriptase protein in the cells. We also analyzed metastatic. First of all, we analyzed the presence of luciferase positive cells in distant organs by ex vivo BLI. For the purpose, organs were excised and analyzed in IVIS spectrum. As you can see here in the lungs, liver, and spleen, we can find the pronounced number of luciferase positive cells, and their number also depends on the level of reverse transcriptase expression per cell. And we also analyzed physical by histopathology analysis presence and number and the size of metastasis in liver, and this is the bottom right panel, and you can see here that the mean number of metastasis in liver increases with the increasing level of reverse transcriptase expression, and it's highly correlated with the RT protein per cell and the level of expression of twist. We decided to go deeper into metabolism, and we analyzed mitochondrial respiration and glycolysis using the Seahorse program by Agilent. As for glycostrase, it was assessed by glycolysis using the 2 different concentration of glucose, using low concentration as physiological concentration and high concentration as it's usually supplemented in the DMIM culture media. And for metastasis, it was assessed using the oligomycin, FCCP, and antimutin A. For glycolysis, we found no significant differences between parental 41-Luc2 cells and all the reverse transcriptase expressing subclones in none of the parameters. None of the glycolysis is low glucose level, high glucose level, glycolytic capacity, or glycolytic reserve. But for respiration, we found that it significantly increased upon RT expression in all generated subclones, and the level also correlated with the level of reverse transcriptase expression in cells. So, as summary, in mammary gland adenocarcinoma cells that stable express transcriptase of clade A HIV, this expression leads to increased level of reactive oxygen species and increased migration capacity associated with the expression of epithelial mesenchymal transition marker expression. And all these parameters depend on the level of reverse transcriptase per cell. In vivo reverse transcriptase expression leads to increased metastatic and tumorigenic potential of all generated subclones, depending on the level of reverse transcriptase protein and level of twist mRNA expression. Reverse transcriptase expression leads to increase in respiration in all generated subclones with no changes in glycolysis, and we hypothesize that this increase increase in respiration is associated with increased level of reactive oxygen species that we that we measured in this subconcept, the more respiration activities, the more electron flow through the electron transfer chain, and the more electron leakage generate reactive oxygen species in cells. That's all. Thank you for your attention.
Thank you very much, Ekaterina. I would like to ask you something, because called me the attention strongly that the incidence of the Kaposi sarcoma is around 800-fold in these kind of patients, no? So is there any, do you think that there's any protein similar to the reverse transcriptase there, or how could you explain that?
We are trying to evaluate interaction between HIV proteins and other proteins, but for now, I don't have an answer for your question. It's we're looking for it, but we have no answer yet. Okay.
You only show this types of cancer, Kaposi sarcoma and non-Hodgkin lymphoma. I guess that also for other cancer types, there is a fall increase in relation with the normal population.
Yes, there is a list of all types of cancers, and this one was typically associated with AIDS, and it's well known as AIDS-defined cancers. That's why I put only 2 on slide, but you are absolutely right that there's a huge list, and all types of cancers increased in the people living with HIV population.
Okay, thank you very much. I think there is no question, so if anyone want to ask something, we have a few minutes.